So the lecture I'll give, I'm going to make some general points about science and the reasons why you should do it. They're my definition of science. You've heard other people. I'm going to talk a little bit about the historical background to the oxygen sensing problem, which has quite a strong pedigree, and I hope will demonstrate to people how science works, how knowledge builds on knowledge in unexpected ways. Unexpected is the important word for those who would like science to have an immediate application. It works unexpectedly. I'll talk about a few moments in the oxygen sensing discovery story, and I hope that this will amuse you. And I'll look at Peter, and if he puts his hand up, that means I've made a joke, and I want you to laugh at the joke. Please. That was a joke in itself. But part of the reason for this is that a scientific journey is actually it's quite anxious provoking at the time. It has all sorts of twists and turns and uncertainties. But in retrospect, it's quite amusing, and I'd like you to enjoy some amusing stories. The reason for telling it in this way is I hope it'll give you confidence that there's no particular difference between you and what I'm going to describe, other than that I've been a little bit lucky. And you should move confidently in your life and aim to be lucky. So, where are my controls? So the first thing I want to set out a few general points on science. So what is science? I mean, we've heard a lot of people say what they think science is, but for many people, science is a very unnatural and unusual thing. It differs from scholarship, which you'll know is important in your schoolwork. Most or perhaps all human societies embrace scholarship, by which I mean they learn from their elders, from the wise people, what the facts of life were. Scholarship, and people became extremely good at that. But it's generally believed that science is different. It was only invented once, unlike scholarship. That was in Renaissance Europe. And what they did for the first time, which is very unnatural, is they did things in order to test whether something was true or false, whether it was supported by the outcome of the experiment or not. And that's counterintuitive. Most of us like to believe things. And of course, we believe things more if they're told to us by wise and supposedly important people. But the reason for explaining that is it will change your way of looking at every problem, not just conventional science problems as they arise in the laboratory. So I want to make that distinction, and I want to exemplify it in relation to the history of the field I'm going to talk about. So what is the relevance of history to science? We're going to do a fair bit of history first, or of science to history. And I will argue first for the politicians and leaders. This is a story. I have 4 children, as you heard. My second daughter was to study history at school, and the schoolmaster was explaining the syllabus for history. And at the end of this, one of the parents put their hand up and said, "Excuse me, what you have described is the history of violence, this war and this concept and so on. Why does my daughter need to hear the history of human violence?" And that had an impact on me. This is a conventional way to teach history. But I would argue that it's actually not violence is not what drives society. Science is above warfare. Science, the scientific advances were what made one nation prosperous, and so on and so forth. So in terms of understanding how your nation might succeed, the history of science is more important than the history of military conquest. What about scientists? Of course, we have lots of facts to learn, but if we want to be a discovery science, and I'm hearing that some of you might, then as well as reading and learning all the things you have to learn, you need to read the history of science. Unfortunately, people who read the history of science are old people like me. They have nothing more to give to science, but they read the history. So you young people should read the history of science because, of course, it's obvious. If you want to discover something, read how other people discovered something, and you will appreciate the chaotic, random, fortuitous way in which the discoveries are made. And then, I put more than 90% of scientists that have ever lived are alive today. Why is this? That's because following 500, 600 years ago, the bringing of the scientific method, knowledge builds on knowledge in so many unpredictable ways. We have so much knowledge, we have so much technology, we have so much opportunity to discover it. But what are the challenges for you? And it was very good to hear credit given to teachers. So, as you heard, I had a call from Stockholm on the first Monday of October 2019. A week later, I took another call. It was from my daughter. She had a little girl. So I know there are more grandparents than Nobel Laureates, but you know, which of them is more important? The little girl. But you know what? The little girl, she knew nothing whatsoever. She knew no science, no molecular biology. She's 6 years old now. Pretty well she knows nothing. No molecular biology, just learning to read and write. So people don't appreciate the significance of this. The human brain must be loaded up from scratch. Of course it's obvious, but people don't see that. So the challenge of teaching becomes ever greater with the acquisition of knowledge. I want to make that point very strongly, and as I said last night, I hope the teachers enjoyed the speech. I can see where I would pay a lawyer and a doctor more than a teacher. That's because I've had my education. I might need some adjustment to my health or my finance, so that's my personal choice. But I cannot see, and this is for the government, why the state would not pay a teacher more than a doctor or a scientist. I cannot see that, because the whole of prosperity demands that knowledge, and it's all based on teaching. The human brain must be loaded up from scratch. And for the students, this is akin to what apps you put on your mobile phone, what you put in the brain. So please pay attention to your teachers, and teachers, please rejoice that you own the future of your society. So I'll give some illustrations of that as we go through, talking about the mechanism by which your cell sends oxygen. And as I point, certain parts of this, I hope, are very funny in retrospect. So oxygen and oxygen homeostasis, how did it begin? This is a little statement of the levels of oxygen on Earth, and you'll see that they rise at about 500 million years ago, caused by photosynthesis. The system that I'm going to describe probably arose about 500 million years ago, at that time when the increase in the Earth's oxygen enabled the evolution of animals. Why is oxygen a particularly important issue in animals? That's because oxygen supplies the energy, and animals are large things that can move, so they require more energy. And that's the problem, a very large animal getting the oxygen in precisely right amounts to the right parts of the animal. In human, just a little bit, there we are. In the human, oxygen homeostasis delivery to about 40 trillion cells. Oxygen deprivation will kill you in minutes, and hypoxia complicates most human disease. So I'm going to argue that the question was quite important. So what about the history? Let's begin at the beginning, because this subject has quite an interesting pedigree, and it illustrates how knowledge builds on knowledge in very uncertain ways. Some people demand the utility of knowledge. They usually are politicians. Some people demand the completeness of knowledge. They usually are journal editors. Neither qualification is appropriate. Knowledge is knowledge. The only qualification is that it's correct. So where did this begin? I'd say this is a schools lecture. I'm usually talking to 20, 30 people, so this is a Q&A. Who is this person who started it all, and what did they do? I'd like you to write the answer on your little note paper, see who's correct. But this is William Harvey, who discovered, or rather predicted, because he couldn't see the little blood vessels, predicted the circulation of blood. And how does it relate? So this is the beginning of the story, actually the first correct piece of internal human physiology. This is one thing he wrote: all parts are nourished, cherished, quickened with the blood. In these parts, that's the periphery, if it were made barren, and then it returns to the heart. So what he did there, he's filling in a little few gaps in the understanding. How did Harvey induce that the blood must circulate? That's the question I ask people. How would you do that? And interestingly, why did it take the whole of humanity tens of thousands of years until, well, I've written 1628, he wrote this up. Why was it so long in coming? Why is it not obvious? So this is the point about science being very unnatural. Harvey did two things. One, he noticed the valves in the veins were in a certain direction, always allowing the blood back to the heart. But he did another thing, which was rather macabre. He cut into the artery of animals and what have you, and saw the huge rush of blood out of the artery, and deduced that the conventional explanation that the blood was made in the liver and simply flowed out in one way to the periphery couldn't possibly be true. There was by 50-fold too much blood for that to be correct. Now, why was that interesting? For centuries, humans had fought each other on battlefields. Thousands of millions of people must have observed the blood come out of a large artery, but no one questioned, no one thought that the conventional wisdom that the blood was made in the liver could possibly be correct. So that's the point. Science, in this case, Harvey called it ocular experiments, simple, careful observation, proved something that everyone believed, all the scholars believed was correct, was not correct. But he had trouble, as I'm going to explain. The knowledge was not complete. People said, well, this can't be true because we can't understand why it goes round and round. Well, that's another false argument, but it's an argument that we commonly meet as scientists, that you haven't given the complete picture, so we're not yet ready to publish your work. Of course, we couldn't understand that. We know now the circulation of blood carries oxygen. Why could we not deduce that at the time? Why was Harvey having to consider these things? So the next one, who discovered oxygen and how? Well, these people are generally credited. Shahrokh, the Swedish biochemist, Joseph Priestley, and Antoine Lavoisier. This was 100 years on. Why was it 100 years later before we could discover oxygen? And the answer is that this knowledge, this question, was only introduced by a previous question. First question was, what was air? Was it something or nothing? And people do Boyle's law. PV is a constant. Boyle discovered that the air is springy, that there's something about it. It's not nothing. It has a certain quality that when you compress it. So that piece of knowledge led to the next inquiry. Was there one type of air or more than one type of air? And that was the next consideration. Different types of air could be made in different ways. And Priestley in particular was an experimental scientist doing rather unpleasant experiments, actually, seeing how long a candle or a mouse would survive in a bell jar. And he noticed that the same types of air were good at supporting both combustion and life, and deduced that they were broadly the same thing. We'd have to give the credit to Antoine Lavoisier for getting the chemistry right, that this process of burning and metabolism were equivalent, chemically equivalent, and that what happened was that the material was combining with something in there that was oxygen. He deduced that by being able to weigh. So that was a technical advance that brought that knowledge, that you weighed the products of burning, they actually increased in weight. But I spoke about the importance of experimentation and how it can deliver unexpected results. And the prize there has to go to Joseph Priestley, who went on doing more experiments than you might naturally have done. And if animal life was supported by this particular gas, oxygen, what about plant life? And he discovered, to his enormous surprise, that it wasn't the same thing, that plants actually purified the air and made it more able to support animals. And that of course, was the start of symbiosis. So we'll fast forward a little bit. 19th century, what do we know? The blood goes round and round the body carrying oxygen. The lungs add oxygen to the blood. The blood changes color when it carries oxygen. The tissues of the body use oxygen to live. This process is basically the same as combustion, as burning, and oxygen is carried by the pigment in the blood red cells. That equivalence of combustion and metabolism, I would say, was the first molecular biology, the first reduction of a biological principle to chemistry. So two firsts there, the first piece of physiology, the first piece of reductionist biology. But what next? Well, a very interesting thing, in the face of low oxygen or different oxygen availability, was the whole thing fixed or did it change? And we'll forward now to the beginning of the 20th century. Paul Berg in the 19th century noted thick, dark blood at altitude. And the question was that genetic? Was it altitude dwellers were different? Or did the body adapt to high altitude? So on the right-hand side of this, I'm showing Jess Haldane's expedition to Pikes Peak, Colorado, where the intention was to make measurements at altitude to understand the physiology of altitude, which was shown to be largely the physiology of low oxygen. But I've circled the woman in that party, Mabel Fitzgerald. So this is quite an interesting story. She was not allowed to become a member of Oxford University on grounds of her gender. The men there are sitting at the top of Pikes Peak, and that place was chosen because it had a nice tourist hotel and a railway line taking them to 4,000 meters where they could do the experiments in comfort. However, the men became uncomfortable that Fitzgerald was unchaperoned at the summit, so they sent her off on her own, on a mule, to tour the villages at intermediate altitudes where she made the measurements that I'm going to argue strode to the field. So this was essentially the Wild West. She must have been a formidable person because she made the very beautiful measurements that I would argue first demonstrated the extraordinary sensitivity of the body's adaptation to low oxygen. So there they are on the left-hand side, those graphs showing that even maybe 1,000, 2,000, certainly 1,000 meters will alter your blood hemoglobin level, and you do so in relation to the amount of oxygen. So this is not a very severe stimulus, and that's what attracted me to the field. The credit for linking these observations to the regulation of erythropoietin being a hormone, a chemical messenger that goes through the blood, to go to Alan Erschlev, who demonstrated the hormonal function of EPO by plasma transfer. So the plasma of an anemic animal was more effective in stimulating erythropoiesis in another animal. So that led me to the problem of how to connect erythropoietin to oxygen, that simple problem that I believed, although people argued it might not be terribly important, I didn't know whether it was important or not. I just believed on the basis of sensitivity and some other observations that it might be soluble, and just might be soluble in my hands at that time. So this is the next little bit. I'll tell you some stories from my own research. So this was our attempt. So you need to know a little bit of molecular biology for this, but I'll hopefully try and keep it simple. What you see there is a drawing of a gene. So that was an opportunity. The erythropoietin gene had been identified, and we could get the encoding the DNA from that locus. And what we wanted to do was use that opportunity of a cloned gene to move outwards from the gene to what we believed would be a specific oxygen sensor. So how would we do that? And what we do is take little pieces from that gene, and you see I've chalked in a black bit there that lies at one end of the gene. These grey bits are the actual gene, and we've linked it to another gene that doesn't ordinarily respond to oxygen, and when we've linked it there, we see that response. So that was the first step in the journey, that we managed to show that that bit marked in black was like an addressograph. It was something that told the gene to be switched on in low oxygen. And all genes have these elements, control elements, that tell the gene that you should be switched on in a different tissue or under different circumstances. What we believed, of course, was that sequence had to interact with something upstream that was sensing oxygen. But then we did the first, and I'm going to explain these eureka moments, the first of the experiments that changed my life, and the reason I'm here today. What would happen if we put that little addressograph into a cell that didn't make erythropoietin had got nothing to do with the cells that made erythropoietin, which are in the liver and the kidney. And what you can see there is we saw pretty well precisely the same thing. The initial thought on seeing this was rather disappointing. Maybe it's some non-specific thing. Maybe we haven't found anything out that's meaningful. And then we thought, well, perhaps we have. And there's another explanation for this, that all cells in the body must have a similar sensitive system that was responding to oxygen. And they must be doing different things. This is a little bit like taking a little postal note in Kidlington, Oxfordshire, where I live, and putting that note in the post and it going to Tesco's and bringing the groceries back to my house. I then post the same thing in Hungary, and it has a similar action. It means that the whole of the world is working on a postal system, and I believe that I was then going to discover many, many other things that were subject to this highly sensitive oxygen sensing system. And I believe this was going to be important. I'm quite an anxious person, actually, so what I did, we have to publish work, and I was anxious to get this published, so on Maunday, Thursday, that's the day before the Easter vacation, I drove in my old sports car to London with a manuscript to the offices of Nature magazine. And my idea was that I would burst into the editorial office and explain the matters, what we'd found, to the editor, and they'd immediately see its importance, like myself. What happened, well, I got to the front desk and there's a rather powerful lady there, and she said, they're all at lunch. It seemed a little bit improbable because it was 10:30 in the morning, but I couldn't get past to the editor to make my case. And of course, this letter had become quite famous. August is 5 months on from Easter, and when we see these letters in science, we always look for the word pleased or sorry in the first sentence or so. If they say they're sorry, that means they're going to reject your paper. If they're pleased, that means they're going to accept it. So we immediately scan the letter for these. And that one, as you say, it says, I am very sorry. So that's really bad news. But the important line I've underlined, it says, it explains the delay. It says, we had difficulty finding reviewers. And for the young people, it's not at all, as we've heard, it's not at all unusual for letters of things that go on to be prize-worthy to have been rejected. But we had difficulty finding reviewers. That means the field was rather small. That is going to make your life a lot easier than if you're entering a competitive field. And indeed, it was a small field, and that was lucky because we weren't massively competent in doing the things we needed to do, and we got through anyway, despite my absence of a biochemical training. So that was the first story. It's a really simple experiment, as the editor pointed out. It's only one experiment, but the referees didn't agree with it. To be honest, I didn't think the paper needed refereeing. I thought the data was clear and the implications, it's obvious. It did turn out to be true that the system we then worked on was actually in all animal cells and does lots of different things. So what about that small field, that difficulty in finding reviewers and the importance of being in your own little field? Well, this is an experiment for a little bit further on in the journey. We were trying to move outwards from that EPO gene to what we thought was an oxygen sensor. And we had to do this. This is a common thing in biochemistry. we realized that this HIF protein, here represented by a peptide, that was the thing that bound that address label and presumably contained the information to be switched on by oxygen. We found that it interacted with another molecule, VHL, for a human disease, von Hippel-Lindau disease, and it's important to realize that medical observation has a very good pedigree in informing scientific experimentation. And it had been noted that people who had VHL disease, von Hippel-Lindau disease, often had higher levels of red cells, and a connection was made with the hypoxic system that we were working on. It wasn't us that made the first connection, but this is what we had to do. We were trying to work out what principle it was which led to the oxygen-dependent association of VHL, von Hippel-Lindau, with HIF, hypoxia-inducible factor. And we were very clumsy in this. It took a long time. It took a lot longer than a competent biochemist would have taken. And we eventually worked out, we made a guess, that the modification was hydroxylation of proline. Now, that's a technical term, hydroxylation, but it refers to just the addition of one oxygen atom to the proline ring. And I'm going to show you this experiment, not because I want you to really know a lot about proline rings, but because this is an emotive part of my scientific journey. And here is the experiment with beautiful clarity. Normally, you see these two lanes. The HIF has to be treated with an extract, so you've got a plus in that column, a minus in that, and then it binds and it captures the von Hippel-Lindau. And you see on the right-hand side, that is not the case, and no treatment with extract is needed, and you'll see that result with total clarity. And people were talking this morning about the things that drive us on in science. This is a eureka moment. This is addictive. This is going to change your life. Once you've had a few of these, you're always looking for the next one, and so I still am. So this is what I call a Sunday morning experiment, a Sunday morning moment. That means that a postdoc rang me at home excitedly with the result on a Sunday morning. It implies that the experiment was conducted on a Saturday, which is a little bit unusual these days. It also implies that the result was considered likely to be of importance, so someone went in and read it on a Sunday morning. That is furthermore unusual. But the really striking thing that you have to be robust to, it means the result was positive. Normally, people read the result, it's negative, and they crawl in on Monday morning and tell you the awful news that your hypothesis has been destroyed. But this is a Sunday morning experiment, and that's, again, a result with total clarity. Why was it important? Because it implied that the modifying principle which led to that hydroxylation of proline was a prolyl hydroxylase, and they were known to be enzymes that consume oxygen. So they were a very good candidate for being the oxygen sensor. But nevertheless, we had another obstacle to cross. What was the prolyl hydroxylase? By this time, the field has become rather competitive. So the next story is the third eureka moment. But this is one really for the politicians here and funders and people who make judgments about the worth or otherwise of science. So what happened? Well, we met with this man, Christopher Schofield. People say it was fortunate. It wasn't fortunate because we rang him up deliberately. The English word would be felicitous, i.e. it was a happy moment, but it was deliberate. And the reason we rang him up, because he knew about this type of enzyme. The reason he knew about this type of enzyme was he was working on antibiotic synthesis. quite a different thing. So who would think that antibiotic synthesis was going to be informative for what we wanted? But Chris solved the structure of an enzyme involved in penicillin synthesis. And below there I've put the outline structure of that enzyme. It has those arrows, beta sheets, and this is what's called the beta barrel jelly roll, or in England Swiss roll, and that's a form of cake. And the cake coordinates certain residues that bind iron on the second and seventh strand there. And that enabled Chris to predict that in the human genome, in the human proteome rather, that had now been identified, there were probably about 60, 70 of these enzymes, and he could say which genes were likely to be this type of enzyme. Now, knowledge builds on knowledge in unexpected ways. Bob Horowitz also went to Stockholm for the Nobel, that he was working on quite a different thing, animal form. And this paper, probably one of the most tedious titles you've ever read. It catalogues 150 or so mutant nematode worms that have difficulty in laying their eggs. So he described different aberrations of body form, and one of them was difficulty laying eggs. They catalogued all the mutants, they made them available, and they were published in this paper. And here is a worm with difficulty laying its eggs. What possible relevance could that be to the problem I was trying to solve? Well, at the time we had an Irish postdoc in the lab, John O'Rourke, and John said, I don't really like, we were mapping regulatory sequences. He said, I don't like that, it's too hard work. What I want to be is a bioinformatician. And I said to John, well, it's difficult because we're not a bioinformatics lab, but if you like, you can see if there is a HIF species, that's what we were working on, in nematode worms, in C. elegans. And he came back and said, well, Peter, I think this sequence might be the C. elegans HIF. And of course we didn't know. How could we know? How could we know? And this is a point for the leaders of your institutions. How could we know that? What am I showing on the left-hand side? It's a Western blot which requires an antibody. So the key here is that you must be able to make your own reagents, make your own antibodies. And we had a system in the institute I was working in that could make antibodies, so we made antibodies to John's protein, his predicted C. elegans. There it is, a beautiful blot showing the hypoxic induction of HIF. So that is a really important point. If you wish to lead a field, you must be able to make your own reagents. By the time you've been able to buy them from other places, the whole world will be catching up. So that was John's prediction. It was indeed a HIF protein in C. elegans worms. So how did this one end? And well, turned out that one of Bob Horowitz's mutants that was deficient in laying eggs, we put it there, EGL9, egg-laying deficient number 9, corresponded to one of Chris's predicted oxygenase proteins. It could be the oxygen sensor, and you can see again one of these addictive results with total clarity. The wild-type enzyme, the wild-type worm induces the HIF with hypoxia. The mutant worm that destroys the enzyme, thereby mimicking hypoxia, has this very obvious here in 3 different alleles, stabilization of HIF. So that's a story, that's all there is to it. Took 15 years, just I've summarized it in what, just over 35 minutes. We have a a regulatory oxygenase. That is an enzyme that uses oxygen, splits molecular oxygen. It puts an oxygen atom on the proline ring. That makes an alcohol group, which the chemistry people of you will know allows hydrogen bonding, and that's the hydrogen bonding to VHL, which is a protein that destroys HIF then in the presence of oxygen. So if you have oxygen, you make that hydroxyproline, you hydrogen bond to VHL. VHL is its destroyer. You destroy the system. Very odd, that. Very odd. Making and destroying, but we often see that, and I'll come to this in the final thing, which I hope you'll think is funny. But of course, we heard about translation, and this simple system has allowed the production of two types of drug, not done by my laboratory, I have to say. This was the extraordinary bravery of the pharmaceutical industry, or at least the shareholders in the pharmaceutical industry, that took the risk to see if you could manipulate this system in humans and use that to treat disease. There are, in fact, two classes of drug, one which inhibits HIF to be used in kidney cancer, which is supported by this system being operated to deliver oxygen, and one which antagonizes the hydroxylation to enhance HIF and correct diseases of low oxygen. And the most important, obvious one was where we started with erythropoietin, and that's a drug-like compound. It's a fixed small molecule that works like a key in a lock, except it clogs the lock up so you can't open it. So it is an inhibitor mimicking hypoxia, and those drugs are on the market to treat anemia. We hope that they will be also used for other types of disease. I said that most human diseases was complicated by low oxygen, so our hope would be that other diseases like myocardial infarction, stroke, we can enhance the growth of collateral blood vessels, that's the ones that come around the side to treat the area, and improve all sorts of diseases. But I said I'd try and amuse you in this talk. What I want to come to, we talked about things being anti-intuitive. And what we do in science, when we want to improve our CV or help each other become better known, we write reviews. People who do science will know you can write a review, it can get published, and it increases your number of citations and what have you. So all sorts of people, including myself, had written reviews as to what they thought the mechanism of oxygen sensing was during the course of the 15 years I've described to you. Every single one of the reviews was wrong. And the more intelligent, the more powerful the author of the review, the more it retarded their field, the more wrong it was. A reasonable person would, every single iron protein in biochemistry book was proposed, except the class of oxygen that does it. Why was that? A reasonable person would have said, well, these people know the answer, and they're deliberately confusing the field by suggesting other possibilities which are incorrect. So why is biology so difficult to discover, and why are drugs so difficult to make? Well, this is to do with Darwinian evolution, which of course is not believed by everybody, but I will argue in this lecture that the implications are not appreciated by most scientists, what it does. So Darwinian evolution uses anything it can find for whatever purpose is under selection. The mutations are random, and the selection imposes utility. So what we have, this I've drawn basic BHLH, PAS, ODD. BHLH stands for basic helix loop helix. This is a HIF protein, and that's the basic helix loop helix bit. It's in all eukaryotic species, animals, yeast, plants. What about the past domain? Well, that's in actually even more primitive species. It's in all species. What does it do in bacteria? It's a sensing domain. What sort of sensing? Oxygen sensing. So why would you not predict the past domain was the basis of oxygen sensing? But I've just shown you the evidence that it isn't. The hydroxylation came along later with these enzymes that are in a range of non-animal and animal species, and they do what I've just described to you. They confer the oxygen sensing. And then actually there's another domain on the end of the protein that does a similar thing, but it doesn't destroy HIF, it regulates the activity of HIF. So where is all this leading? You're pasted together by all sorts of bits in a very illogical way that makes our job as scientists quite difficult. To be clear, it doesn't help to be very clever in biological discovery research. It might be a hindrance. And I'll explain why this is by reference to the European motorway system. That's a joke, Peter. And I'm not going to talk about the Hungarian motorways, or let's just deal with we'll deal with Germany first. We're on the Autobahn and we're traveling south. Stuttgart is on the right of the Autobahn. You look on your Mercedes, there's the sat-navs, there's Stuttgart to the right, 500 meters. And as you get close to the junction, you will see a big sign to Stuttgart. To the right, and the Mercedes you drive to the right, where will you go to? Stuttgart, obviously. Where is this? Where is this place? Can anyone tell me? This is not Germany. It's not Hungary either. It's not Germany, it's not Hungary. It's not the United Kingdom either. We have the good fortune to have a house in the south of France. And this is France. This is the south of France. And what you have Nice up here, and you have Cannes here, and down here we have Juan-les-Pins, and that's where our house is. So if you wish to go to Juan-les-Pins, you come down the motorway, you come off here, you go up here, you go around the woods, you go down here, you go under the motorway, over the motorway again, round this little bit, you turn again, and eventually you get to Juan-les-Pins. Now, this is different from Germany, you see. Your sat-nav is going to be confused. Your partner will attempt to give you instructions. You will become very angry with your partner. Rational thought will not prevail. And the only way that you're going to understand how to get to Juan-les-Pins is trial and error. And this is how you're made. And what is the crux of the story? Well, people say that in France, particularly in the south, the French have better things to do than build roads. Better things to do than build roads. So what do they actually do? Well, and how do they arrive at something like this? Well, they take pieces of existing road and they merge them together to make the roads they want. And that is Darwin and evolution, as Francois Jacob said it so beautifully. Nature is a tinkerer, not an engineer. And the problem we have with translational medicine, which is a problem, is that the drug designers aspire to be engineers, not tinkerers. And that is, I'll leave you with that. That is one of the most important restrictions in what we have to do. So why should you study science? Well, we'll be a little bit arrogant here, because it's important. What do I mean by that? Well, you can change the world. The most satisfying thing about what I've told you is, well, it was done by intention, is that this is true, this sensing system I've given you. It was probably true 500 million years ago at the origin of animal life, and if there is ongoing life for 1 million years, it will be true then. Knowledge cannot go backwards. So that is extraordinarily satisfying and addictive and important. For the children here, you can think of Julius Caesar, you know, Veni, Vici, came, I saw, I conquered. What is the impact of Julius Caesar on current knowledge? You know, close to zero. So even the most famous leaders do not reach eternity, but your knowledge will. So it's important because, as I hope I've illustrated, at times it's fun. Because you're living in an age of opportunity when there is more technology, more existing knowledge on which you can build, and you need your teachers to support you in that. And because, as I have tried to argue, science is different from scholarship, your natural tendency is to be a scholar, not a scientist. Think carefully about those experiments that prove or disprove by observation that something is true or false. It will probably change the way you think about anything you choose to do with your life. So I'll leave you with that. These are the people who've helped me recently in the work. This is Oxford on a nice sunny day. And thank you very much for your attention.
Stay there, and thank you very much for this lovely presentation. And now it's question time. That I don't know that here we will have microphones. So anybody would like to ask a question, here you are allowed to ask. I don't know any questions from the floor. Raise your hand. Questions? Maybe there is a question over there. Yeah.
Thank you. So it might be a strange question, I'm sorry, but as a Nobel Prize winner scientist, what is your opinion about the relationship between religion and science? Because a lot of scientists only believe either one of them has to be correct, but I'm interested in your opinion.
So, can I switch the microphone on? Yeah. Well, interesting. I'm not going to answer the question directly, but I will make one observation. I'm a fellow of the Royal Society, which is a science society. And my observation there is, yes, what you say is true, that the biologists tend not to believe in religion, and I don't know. I mean, Darwin produced a theory of evolution for which the evidence is extremely strong. Of course, he talked about the origin of species, not the origin of life, which is an interesting thing to talk about. The physicists and mathematicians at the Royal Society are more inclined to believe in religion than the biologists. I'll just leave you with that observation that I've talked about prejudice. I'm not going to be drawn into a prejudice that biology is always right and there is no place in the world for anything other than scientific thought as we see it. That can't possibly be true. But our observation is that embracing these things is much more common in theoretical physicists than it is in biologists, and they're all scientists.
Thank you. Other questions? Yes, there is a question over there.
Thank you very much for the fascinating presentation. I only have one question regarding the scientific methods. that you showed in the presentation. So did you have to do the experiments in different cell types, or did you only focus on one cell type?
No, that was the point of that experiment, that we did it in different cell types, and in fact in all cell types that we studied, in human and mammalian cells, and indeed in all animals that we've studied, we found the same phenomenon. That's correct. But it does different things because the cells don't make erythropoietin.
Thank you very much.
Other questions?
Thank you for the presentation. I really enjoyed it. I have a question. So, why do you think the HIF pathway is so strongly conserved across animals despite the wide differences in oxygen environments? Because you mentioned how blood and oxygen is different. It basically changes with altitude.
Yeah, that is a very interesting and informed question that we've thought about a lot. Probably has to do with Darwin and evolution again. So once you've evolved a certain mechanistic system, that's what you're stuck with. So an animal, despite the fact that it would obviously be faster to run on wheels than legs, the cheetah does not run on wheels, it runs on legs. So we have evolved this system which has multiple components, the HIF target genes, the sensor. Once you've done that, the easiest way to evolve is to add another little bit on that, like the French designed their motorways, rather than scrap the whole system and start again with a different type of oxygen sensor. But what you say is extremely interesting. The nematode worm oxygen sensing, if I take your sensor out of the human cell, put the worm in, it will work, despite the fact that the worm's in a completely different environment. And I assume there are adjusters that tune that central system to make it into one that's fit for another type of animal. So what I'm saying is most of evolution is about adjustment, about tinkering, as Jakob proposed, rather than re-engineering the system. And that's another example of it.
Okay, other questions? It's over there.
Thank you very much for
I didn't hear the question.
So thank you very much for the awesome presentation. I would like to ask your opinion regarding intuition and the role of accidents in research. So when you are practicing in a research group, and what's the main role behind the great breakthrough? It's something regarding an accident or intuition or a well thought of and calculated thought process. And what's more valuable to you? Something from intuition and or an accident, or more like from well calculated and thought process?
I don't know really. I'm a clinician as well, so I'll give you one difference between clinical medicine and what we do in the lab. In clinical medicine, most doctors will know that if you don't know what to do, it's a good idea to do nothing at all. Right? In the lab, that cannot possibly work. So in the lab, if we don't know what to do, we should do something. We should do the best experiment we can, because nothing will happen. The reason for that is, of course, clinical medicine is evolving before your eyes anyway, whereas if you do nothing in the lab, you will gain absolutely nothing. So I think the important rule is to be as unprejudiced as you can and do experiments.
Thank you. Other questions? Okay.
So my question would be, what was the main reason why you got up every day and started it again, even if you failed the day before, and what was the reason that encouraged you along this journey?
Well, I think it's just fascination for finding things out. To be clear, well, it might seem arrogant, but the reason that I showed you that letter was that we preserved that letter. There's an implication in that we thought we would. We thought we were onto something, so it was not really necessary to sustain, a period of disbelief. For a long, long time, I believed that this problem was soluble, and it might not be soluble by my group, but it would be solved by someone. And when we got that letter, we filed it away, and we thought, oh, that's interesting, they don't want the paper now. Maybe it'll come up in the future. You might say that was arrogant, but actually we were not that greatly disappointed.
Thank you, and I saw there somebody also wanted to ask. Students, yes.
So you said evolution tinkers and doesn't engineer, and my question is, could we approach scientific methods from this point of view and try tinkering instead of straight-on approaches in engineering?
Well, possibly. I think one of the types of experiment that is very informative is to rerun Darwinian evolution. You can do it two ways. Subspeciation would be very important. So different species of fish or bird that work in different oxygen environments could give very informative answers as to the nature of the systems. Or we could do it deliberately by making mutations and screening for different phenotypes. I mentioned at the start homeostasis, or I mentioned in the earlier talk homeostasis, and how that's probably the biggest unsolved problem in biology. So phenotypic using genetics and connecting it with physiology in an unbiased way is what Darwinian evolution has done. Random mutations selected by physiology, and we can recapitulate that in phenotypic screens.
Thank you very much, and I think this was the question time, so I also would like to ask András Svarrod, the curatorial board, to give you a memory from this event, also a bronze medal. It's not as shiny as the Nobel one, but we really give it with pleasure, and we are very thankful for you being with us.